let's see. What are we doing today? That's not right. I forgot to update that. What we're going to do today is we are going to talk about a little bit about glycogen metabolism, and then we are going to glycogen metabolism, and then lipid oxidation. We're going to tie back into energy metabolism, and we're going to talk about lipid degradation. We got the homeworks due, okay, this last time like this. Okay, for the final exam, okay, we're going to go through, you can bring pathway maps, okay, for the pathways. And part of this is because I'm having to kind of manage the amount of detail that I'm getting into all these pathways, right? And so basically, you can make a pathway map for the glycogen metabolism we're going to talk about today, for the lipid metabolism we're talking about today and tomorrow, and the little bit of extra amino acid metabolism. You can make pathway map for that. You'll turn it in. I'm not going to give you any points for it, but at least you can have it for the exam.
I have a question.
Yes.
Do we also get to use the pathways for the previous things? Or
No,
just
but what I'll say for the comprehensive part of that is I'll be looking at more like overall questions rather than what's this enzyme, in this path, the third enzyme in that pathway kind of thing. Yeah. Zoom help session. What's better, 4 o'clock or 7 o'clock?
7.
7? Okay, I'll set it up for 7. And then, yeah. Any questions about this?
Professor?
Yes.
Are we going to get our pathway map from the last exam back?
Yes, I will give that back to you. I'm hoping to get that done all back to you tomorrow so that then I don't have too many pieces of paper. And you guys know where you're at before. It's not helpful for you guys to know where you're at before going into the final. Any other questions? Okay, I did want to, because I kind of messed up my slide presentation on last Thursday and didn't have a couple of slides. I did want to show you a couple of slides here. This is there we go. This is rubisco, the most abundant enzyme on Earth, the enzyme that's responsible for fixing carbon, bringing fixing in. The one that's kind of weird because it has both the carboxylase activity and the oxygen activity. The other thing that's kind of cool, structurally it has L8S8, so that's the terminology that's historically been used. 8 large subunits, 8 small subunits. Kind of interesting thing is that the large subunits, the chloroplast, like the mitochondria, has its own genome. And so remember when we were talking about the complexes of the electron transport chain with these big complicated structures, and the fact that some of the components within those structures are encoded in the mitochondrial genome and translated within the mitochondria. Others are encoded in the nuclear genome, translated in the nucleus, and then imported, and then all have to be assembled. Same kind of thing. The small subunit is encoded in the nucleus, and so it's translated out in the cytoplasm, has to be imported into the chloroplast, and the chloroplast has its own genome. It encodes the large subunit, and it has to be all assembled. And again, it's also a highly regulated process. So that whole Calvin cycle, all those enzymes are really only on during the light when the photosynthetic electron transport chain is making the ATP and the NADPH that this cycle needs to be able to work. We'd gone through this production phase. I won't redo this, but the other aspect of it is the end of that phase, the reactions that I talked about. We talked about glyceraldehyde 3-phosphate. That was sort of the product, and I basically put 2 arrows. Okay, it can be used in gluconeogenesis, or it can be used in to remake the ribulose 5-phosphate, and this is, so on top, that's those series of reactions. So this is this is the pathway for the remaking of the ribulose phosphate. You see it's pretty convoluted. You take this 3-carbon molecule and then you take a 3-carbon molecule and you make a 6-carbon molecule, and then you take a 6-carbon molecule and make a let's see, there's a 3 carbon, a 6 carbon, I gotta remember. Yeah, a 6 carbon molecule plus a 3 carbon molecule, and then you make, I can't remember, I lost it all. But basically, there's a bunch of carbon moving around through all here, and eventually you get the rearrangement, and you make that. Now, remember, in this production phase, we fix 1 carbon in here. So you basically, if you run this whole reaction 6 times, then you will, the 6 times will generate 1 glyceraldehyde 3-phosphate that can go to gluconeogenesis, and then the other 5 can be used to remake a bunch of this. And so that you're basically running this whole cycle, and then it just occasionally siphoning off a little bit of glyceraldehyde 3-phosphate to make sugars. So all this cycle was figured out by Melvin Calvin at Cal, and using a lot of radio tracer type experiments where you put radio labeled intermediates and then follow where the label goes. Yeah.
Does Rubisco have more than 1 active site on it? I'm just wondering,
I can't remember. Like in the
so big,
LASA,
does it have 3 sites?
I can't remember.
Okay.
I can't remember. All right. Anyway, I just wanted to, you're not responsible for this, but just kind of get a little bit of appreciation for that. All right. Now let's go to glycogen. Remember glycogen? Glycogen was, the polysaccharide, the branch of alpha-1,4 and alpha-1,6 linkages. We talked about a couple of different roles, talked about the role of it in the liver, because after you've had a lot of dietary carbohydrates, the liver will store away those carbohydrates as glycogen. Then when it's been a while since you've eaten, it'll basically mobilize that, dump it back into the bloodstream, make sure that the blood has enough glucose going around. In muscle cell, it's a little bit different. It'll store away glycogen, and then it has a ready source of carbohydrate when the muscle needs to contract readily and get going. So what we're going to talk about a little bit is glycogen synthesis and glycogen degradation. And we'll start with glycogen degradation. And what I'm going to do is I'm going to draw up the non-reducing end of a glycogen molecule. Remember, we have an alpha-1,4 linkage. So this is the non-reducing end of glycogen. And so we're going to have to cleave this bond, this glycosidic bond that we made when we put the glycogen molecule together. Now, generally, when we cleave monomers from a biological polymer, it's a hydrolysis reaction. So, for example, when we had, you remember back when we had protein and the chymotrypsin mechanism and all of that, we cleave the peptide bond by adding water to that peptide bond to cleave it. Similarly with nucleic acids, if you take a nucleic acid and you chop it up into individual nucleotides, you do a hydrolysis reaction on the phosphodiester backbone. In this case, we don't. We actually do what's known as a phosphorylasis. We actually bring in phosphate, inorganic phosphate. I'm just going to put that as PI. and we generate glucose 1-phosphate. This enzyme that catalyzes this reaction is known as glycogen phosphorylase. So this is a phosphorylasis. And so you're gonna make this plus will I guess I should, sake of completeness, I should fill out all my hydroxyl groups here. This enzyme requires a covalent, requires a coenzyme known as pyridoxal phosphate. Which is vitamin B6. I think I might have mentioned this one. We were talking about various different enzyme cofactors. It has this structure. That's the structure of pyridoxal phosphate. And the reason I went ahead and threw up that structure is because you'll see this cofactor used in other reactions, particularly a lot of reactions in amino acid metabolism, because it contains an aldehyde. And I think I made the point that no side chain of an amino acid has an aldehyde, and that's probably because it's so too reactive and that it would just bugger everything up. But occasionally an aldehyde is useful in chemistry, and so the system deals with that by having this cofactor with the aldehyde functional group. Now it turns out in this particular enzyme, the aldehyde isn't what's important, it's the phosphate that's what's important. But in other enzymes that use pyridoxal phosphate as a cofactor, it's actually the aldehyde chemistry that's required. And so this is glucose 1-phosphate. This is also an important regulatory enzyme. Well, we're not going to be able to get into the detail of it, but just like with glycolysis and gluconeogenesis, there's coordinate regulation of glycogen degradation and glycogen synthesis. And actually, that coordinate regulation is also coordinated with glycolysis and gluconeogenesis. All right, so the last step is we have our glucose 1-phosphate. And to get it to an intermediate, yeah.
Sorry to interrupt. Is that an N attached to an N, or is that just an NH?
That's an H.
Okay, thanks.
So we have glucose 1-phosphate. The next step for it is we can just move that phosphate from the 1 position over to the 6th position, and then now it's something we're familiar with. That's an intermediate glycolysis. So this is now glucose 6-phosphate. Again, this is, this second intermediate of glycolysis. So now you can basically have a couple of different fates of this. If it's a muscle cell, and you're mobilizing your glycogen to be able to get glucose to run glycolysis in the PDH cyclic to be able to generate ADP, you would go this direction for more, you basically do glycolysis on it. But maybe you're a liver cell where your role is actually to maintain blood glucose levels. In that case, you would take the glucose 6-phosphate and make glucose to dump back into the into the bloodstream to maintain blood glucose levels. Now I will introduce you one other step that we kind of ignored. So we notice, again, we started this from the non-reducing end of glycogen. And we've cleaved off of sugar from the non-reducing end of glycogen. And I tried to make the point that one of the sort of rationales for that branch structure of glycogen is because then you have many non-reducing ends and you can rapidly mobilize a lot of glucose if you have a lot of non-reducing reducing ends. But the other aspect about that is, what about the branches? What are we going to do about the branches? Now what I'm going to do is I'm going to draw a glycogen molecule with, except each of these little beads is a glucose molecule. And we have a branch. So we might have glycogen phosphorylase basically chews down the end, but it chews it down until there's about 4 glucose units left. And so 1, 2, 3, 4, and then maybe this has been chewed down a little bit too. This is known as the limit branch. And it won't chop anything down beyond here. I don't know if there's steric hindrance or what it is, but basically the glycogen phosphorylase will chew it down, chomp, 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 until it gets to 4 and it'll stop. So then there is another enzyme known as the debranching enzyme, which will take, I'm going to mark these with a little X just so we can differentiate it. It'll take these 3 and it'll transfer it to this end. And this enzyme is known as a debranching enzyme. So we've just basically taken those three. We've left one here. We still have that one alpha 1,6 linkage. Now, the phosphorylase can access this and chew this down. And what about this little branch here? There is a alpha 1,6 glucosidase that just brings. This is actually a hydrolysis reaction. Just brings water in, and then you get that plus the one sugar, one glucose molecule. Oh, I forgot to give you the name of this one. Here. It's moving the phosphate from the one position to the, this is phosphoglucomutase. Remember we had a phosphoglyceromutase in glycolysis that took the 3-phosphoglycerate and moved to phosphate from the 3 position to the 2 position. So that was phosphoglyceromutase. We also have a phosphoglucoisomerase, which converts the glucose 6-phosphate to the fructose 6-phosphate, but that's rearranging the carbon skeleton. Since here we're just rearranging the phosphate, it's a mutase. And it's a glucose structure, so it's phosphoglucomutase. All right, very good. Now we've taken glycogen down. Now let's build a glycogen molecule back up. And where we're going to start is we're going to start here at glucose 6-phosphate, and then we're going to remake glucose 1-phosphate before we go on to polymerize the glucose. Actually, so what I'm going to do now, let's see. Okay, so this is glycogen synthesis. So in the liver, this is going to happen after you've had a nice carbohydrate-rich meal. There's plenty of dietary carbohydrates. The liver wants to take it and store away this dietary carbohydrates. And muscle cells too, when you have available serum glucose levels and you're not actively contracting, you want to store away some glycogen, muscle glycogen, to be able to have a store of glycogen for the next time you need it. And so we're going to start with our glucose 6-phosphate. So again, I'll just kind of remind you where we are. Here's glucose, like this, and then we have glycolysis going this way, like glycolysis going this way. And now, this is basically a reversible reaction. We're going down to glycogen down here, and then we're going to do the phosphoglucomutase reaction and make glucose 1-phosphate. Nope, this is H, and this is the phosphate. So essentially, really what's going on is, in that glucose 6-phosphate is really, there's a pool of glucose 6-phosphate that's interacting with a pool of glucose 1-phosphate. And if you actually remember the next step in glycolysis is phosphoglucoisomerase, so it's the fructose 6-phosphate, which is all very reversible reactions. So they're basically all interconverting among each other. And then you can basically draw from those different pools. So we have glucose 1-phosphate. Now what we want to do is we want to polymerize this, right? We want to take this and we want to add on to a pre-existing glycogen molecule, add on in the non-reducing end, right? We want to create a glycosidic bond. Now what we have here is we have a phosphate ester. Now the free energy in a phosphate ester just isn't enough to drive the formation of the glycosidic bond. We need a higher energy intermediate. We have to activate that glucose 1-phosphate. So I'm going to redraw. Here's our glucose 1-phosphate. Glucose 1-phosphate. Now what we're going to do is we're going to react this with a nucleotide triphosphate. ATP is a nucleotide triphosphate. We're going to react it with a nucleotide triphosphate. That nucleotide is not ATP but UTP, so uridine triphosphate. And so let me kind of draw, I'm going to start doing an abbreviated structure of uridine diphosphate. So here we've got the ribose, and then we've got 3 phosphates in here. What we're going to do is we're going to create a bond between this phosphate and this phosphate. So we're going to create a bond right here and link this phosphate, and this stuff all to the glucose molecule. Leaving these two will leave. So what we do, we come off with pyrophosphate, PPI, and we end up with if I wanted to be real complete, I could put these hydroxyl groups on here. This molecule is known as UDP glucose. That is activated enough that it can drive the formation of the glycosidic bond. Now, one of the reasons that this reaction is able to go forward thermodynamically is this pyrophosphate molecule will be hydrolyzed in a subsequent reaction to make 2-phosphate. And this enzyme that hydrolyzes this is pyrophosphorylase. So that product of the reaction gets rapidly consumed. That rapid consumption of this product helps drive the thermodynamics of this whole reaction forward. I should give the whole name of this enzyme here. This enzyme is known as UDP glucose pyrophosphorylase.
Professor?
Yeah,
What does that other thing say? Pyrophos?
this right here.
Yeah.
Pyrophosphorylase.
That's the 2P?
What's that?
That's the 2P?
Yes, so that's what it does, is takes this pyrophosphate here and it just hydrolyzes it to the 2-phosphate. And that's a very thermodynamically favorable reaction because you're getting rid of a phospho and hydride bond here. So the fact that this goes so quickly basically sucks that reaction forward.
So the two P's bonded together, or the two phosphates bonded together is the pyrophosphate?
Yes, that's exactly it.
And then this-
Pyrophosphate is an old-fashioned word for diphosphate.
Okay, and then two phosphates just meaning they're separate from each other?
That's right.
Okay.
Here, I'll just do.
No, it's all right, I understand now.
It's basically, one H on there someplace. So we're just splitting that apart. Because this is a phosphoanhydride bond that we're actually cleaving, that's a pretty thermodynamically favored reaction. Okay, so now we have this activated form of glucose with this big thing attached, right? But that, we've got basically a lot of energy available in that, so we're gonna take advantage of that to drive the formation of the glycosidic bond at the non-reducing end of the glycogen molecule. So we now take, here is our so this is the non-reducing end of a existing glycogen molecule. So this is going on and on here. And now I'm not going to redraw that whole structure. I'm just going to say UDP glucose. UDP glucose plus that is going to what we're going to end up now is generating the new glycosidic bond plus UDP. Okay, so now we can add on sugars in there.
Do we need to know the enzyme?
Yes, this is glycogen synthase. So glycogen synthase and glycogen phosphorylase are sort of the counterbalancing reactions in glycogen synthesis or glycogen degradation. Glycogen phosphorylase is the degrading enzyme, and glycogen synthesis is the synthesis enzyme. So those are the ones that are very tightly coordinately regulated. So only one is on when the other one is off, and then vice versa. And in the liver, it's also tied to gluconeogenesis, so the whole signaling cascade basically will, gluconeogenesis is stimulated at, gluconeogenesis is stimulated at the same time glycogen degradation is stimulated, so that both of those processes are generating glucose to be able to dump back into the bloodstream.
Professor, how many phosphates are on the UDP?
DP2. So there's like 1, 2, so this is UDP.
Okay.
Yeah. So what ends up having to happen energetically to like redo another one is you're gonna have to add a phosphate onto that. So that's gonna cost you an ATP to
Gotcha.
remake this. So that's where the energy is basically coming from. But, you know, one other thing I want to mention, I probably got rid of something I just kind of about the whole phosphorylases. I made the point that in degradation you end up, when you break this bond, you're doing it with a phosphate, with a water. And one of the reasons for that, I guess, is that by doing that, you can serve that phosphate ester in there, and then you can just rearrange it over here. If you hydrolyze this bond, you would end up with glucose. Then it would cost you an ATP to remake this phosphate in here. So by doing a phosphorylasis rather than a hydrolysis, you actually save yourself an ATP. All right, so I think that, actually let's go ahead and finish up. So glycogen synthesis, we basically have the phosphoglucomutase, we have the UDP glucose pyrophosphorylase, and then the glycogen synthase, those three reactions. The third one that I'll throw in there is what about those branches? So we basically now have let's see, what was the name of the enzyme that dealt with the branches in glycogen degradation?
Debranching?
Debranching enzyme. Debranching enzyme. So guess what the name is, the one that puts the branches in there?
Branching
enzyme.
enzyme.
Occasionally it actually makes sense. Okay, so how does branching enzyme work? Well, basically maybe you've created, I won't talk about how the first branch comes in. I'm not sure that's, I actually know that. But, okay, so say, you've gone through this glycogen synthesis, you made these long chains, right? Now what the branching enzyme will do, will take, it's usually about 7 of them. And then it will move them
The enzymes are the guys with X's in them?
What's
No,
that?
those are the glucose molecules that get moved.
we're gonna take these and they're gonna just basically, so these here will get transferred to this end here. So, no, not to that end, will get transferred to an edge here to create a new branch. And now these guys can be extended out, and then you can repeat that process, and et cetera, et cetera. So this is known as the branching enzyme. All right, so that kind of finishes up glycogen synthesis and glycogen degradation. What else was I going to say about this? Oh, there's an analogous series of reactions in the biosynthesis of starch in plants. There's a little bit of variation. Instead of UTP, it's actually ATP is the nucleotide. It's called ADP glucose pyrophosphorylase instead of UDP glucose pyrophosphorylase. But it's all very similar. There's branching enzymes like this. And that have been studied in quite a bit of detail just because of the importance of starch quality. And, the starch, especially the cornstarch industry, dries a lot of, you make cornstarch, not all of it's used for animal feed, it's also used to generate high fructose corn syrup. Yeah, David.
If the branching enzyme constrains the outermost glycogen polymer branch,
Is it constrained to the outermost?
yeah, so if this were hypothetically 3 branches long, you have one up, one down.
Oh, I see what
Would
you're
be
saying.
unable to interact.
No, I don't think so. I don't think so. I think it's going to be, because actually these molecules are, remember, are building in 3 dimensions. They're actually spherical. And so there's a lot of this stuff going all around the sphere. There's this stuff, these transfers going on. So what I was going to say about starch, so cornstarch is used in all sorts of different technologies for fermentation, for producing high fructose corn syrup and all that sort of stuff. And you can get variations in starch quality that'll make that process a little bit more efficient and less efficient. And a lot of those variations have to do with mutations in these various enzymes involved in starch biosynthesis. And it's one of the things that people do is they, through either breeding or genetic engineering, try to tweak the different enzymes to make slightly variations of starch structure that is more amenable to whatever that downstream use of that starch is. The other thing, though, the more more major genetic mutation in corn that creates a whole new product is mutations in, and I'm trying to remember exactly which enzyme, but basically results in no production of cornstarch, and then so what the corn kernel does is accumulate sucrose rather than cornstarch, and that's sweet corn, the corn on the cob that we eat. That's basically a mutant corn that can't make cornstarch in the kernel, and makes it just has it accumulate sucrose instead. All right, one other thing before we take a break here. And there's one other pathway that involves this glucose 6-phosphate. So just a little reminder, we have glucose, and then we have the hexokinase reaction, and that's going to make glucose 6-phosphate. And so far we've talked about a couple of different directions. It can continue with glycolysis. Then we've talked about how it can go down to glycogen, or glycogen get reconverted to glucose 6-phosphate. Of course, we also have gluconeogenesis going back the other way. I'm going to throw in one more pathway that involves glucose 6-phosphate, and I'm just going to really summarize what that pathway does. And that's the pentose phosphate pathway. Okay, and then what it's going to make is pentoses like ribose. I'm going to say 5 carbon sugars. Like ribose. So that's ribose is important in the phosphoribo, the nucleotides in the DNA. It's important for DNA. We talked about ribulose being important for in the Calvin cycle in plants. So if you need to make 5 carbon sugars, and in the process of doing that, one, you get rid of a CO2, and you take NADP, and you make NADPH. This is the same thing that is made in photosynthesis, but if you're a non-photosynthetic organism, right, then you need a source of NADPH. And this is a good timing to talk about this, because when we get into lipid metabolism and lipid biosynthesis, we'll see this is required for anabolic pathways, such as lipid biosynthesis.
So Professor, are you saying this happens in us too, not just plants?
That's right.
Okay.
So this is the way that heterotrophic organisms basically get NADPH. That's the pentose phosphate pathway. So you kind of see the glucose 6-phosphate ends up being a kind of an important key intermediate with a lot of different branches.
Is that reversible also, or is that a?
No, that's pretty much a one-way deal.
Okay.
Yeah. And skipping over, remember at the very beginning I showed you the recovery phase of the Calvin cycle, and there's like all these rearrangements that I couldn't actually follow and remember exactly what were going on. Like this pathway actually has a very similar thing. And evolutionarily it's thought, remember that photosynthesis from an evolution standpoint precedes all this heterotrophic metabolism. And so this pathway is thought to have been basically derived from that Calvin cycle, evolutionary derived from that Calvin cycle. Okay, we'll stop, we'll take a break there. We'll come back at 11:30, and then we'll start talking about lipid metabolism, fat metabolism.